Okay, in this video, we're going to talk about photosynthesis, specifically the light reactions of photosynthesis. And in the next lecture, we'll talk about the Kelvin cycle, or sometimes called the dark reactions. So the light reactions are also called photophosphorylation. And here's the ultimate summary of photosynthesis. We need to compare it to respiration. Respiration is when we are taking a reduced molecule like glucose and we are oxidizing those carbons, right? So here we have the carbons. Each carbon is bound to a hydrogen, two other carbons, and an oxygen. Here, all of those carbons are now double bonded each to two oxygens. So that's about as oxidized as you can get, and indeed those carbons of carbon dioxide are excreted. Now, where do those electrons and hydrogens end up? Well, they end up in oxygen to become water. Now, each one of those redox reactions was exergonic, and therefore those that energy was released from those exergonic reactions was used to make ATP. Now, take a look at photosynthesis. Photosynthesis is doing the exact opposite, which means in order to get photosynthesis to go, we're going to need to invest a whole bunch of energy. And the source of that energy is, again, through ATP. If we created ATP via respiration, we're going to need to invest ATP for photosynthesis. So one of the questions that you have to ask yourself as we go through here is, where does that ATP come from? And the answer is that it doesn't come from the mitochondria. Now, don't forget, plants have mitochondria, but the ATP that's made in those mitochondria is going on to do all the various cellular work. The ATP that is made for photosynthesis comes from the chloroplasts themselves in the, as it turns out, as the light reactions, and we'll look into that in a minute. And now we have our oxidized carbons that we are, instead of oxidizing them, we are reducing them. We are attaching more hydrogens to those carbons. We are adding more electrons to those carbons. Where are those hydrogens coming from? Where are those electrons coming from? Well, they are coming from water. So if water is the reduced form of oxygen, that means it has electrons and hydrogens that we can use. And that's what we're going to do. That's going to be our source of hydrogens. That's going to be our source of electrons. Where's the source of carbons for this, this reduced glucose molecule? CO2. All right, so photosynthesis then is the act of stealing electrons and hydrogens from water and giving them to CO2s to make glucose and then our oxygen as a byproduct. All right, so now what we need to do is start to think about how this is working. So the big problem, the ultimate problem here is, remember, oxygen was the ultimate receptor of electrons, because oxygen in animal cells was the most electronegative molecule. It had the ability to steal electrons from any other molecule within the electron transport chain. And yet we are saying that water, the reduced form of oxygen, is the source of electrons for photosynthesis. Well, how can that be, right? Oxygen holds on to electrons far too tightly, especially to be able to donate it to a molecule like NADP+. Now remember, NADP+ is much like NADH, except NADPH is also is involved now in biosynthesis instead of oxidation. And so like NAD+, NADP+ does not hold on to electrons very well, certainly not any better than water, than oxygen. So how is that happening? Well, that's where the sun comes in. The sun provides the energy that allows the NADP+ to steal electrons from water. Now, that's not a direct relationship, and we'll get to that in a second, but that's really it. It's the sun that's going to allow us, for molecules that normally wouldn't be able to strip molecules from water, that suddenly they can. And again, it's not direct. We'll look into it in more detail in a moment. And then that NADPH, that's going to be our source of electrons and hydrogens for the synthesis of glucose from CO2. And this ATP that's being made is also the source of energy for the production of glucose from CO2 here in the dark reactions in the Kelvin cycle. All right, let's start talking about this in a little bit more detail. Now, photosynthesis takes place in chloroplasts within eukaryotic cells. Chloroplasts have an outer membrane and an inner membrane, and then in between the two is the intermembrane space, so much like a mitochondria. However, in the mitochondria, that intermembrane space is very important. Here in the chloroplasts, that space isn't very important. Instead, then we have another set of membranes called the thylakoid membranes that form these discs. So now we have two new spaces. We've got the stroma, which is within the inner membrane, but outside of the thylakoid membrane. So all of this space, this gray space here. And then we have the thylakoid lumen, or the thylakoid space. Now these are the two spaces that are the equivalent of the matrix of the mitochondria and the, intermembrane space of the mitochondria. And so we'll look at those, the comparison a little bit later. Now, photophosphorylation, or the light reactions, like the electron transport chain of mitochondria, is dependent upon enzymes that are embedded within a membrane. In this case, they're embedded within the thylakoid lumen. They're embedded within these green membranes here. There is photosystem 2, there's cytochrome Bf, and there is photosystem 1, and then there is ferredoxin reductase. So 1, 2, 3, 4 enzymes. There are a series of movable electron carriers that are reduced and oxidized. There is Q, or actually in plants it's called PQ, or plastoquinone. There is the plastocyanin, which is the equivalent of cytochrome C, and there's ferredoxin here. Okay? In addition, like the electron transport chain within mitochondria, there is ATP synthase. And so what you're seeing here is there is an evolutionary relationship between what's happening in the mitochondria and what's happening here in the chloroplasts, in that we have a series of redox reactions occurring where one molecule is being oxidized and the other one's being reduced, and that exergonic reaction is being used to pump protons into the thylakoid lumen. That proton gradient is then being used via ATP synthase to make ATP. Now, that means we have an accumulation of ATP here within the stroma. That means the electrons that we are stealing from water and taking to NADPH is also accumulating within the stroma, and it's those two that are going to be the source of energy and the source of electrons and hydrogens that are going to be used to turn carbon dioxide, or 6 of them, into a molecule of glucose. So here's the problem. In the mitochondria, we started with NADH and we ended with oxygen. In photophosphorylation, we're starting with water and ending with NADPH. The problem is, this NAD+ does not hold electrons any better than water does. In fact, much, 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 much worse. So how is it that we get electron flow occurring in the opposite direction? The opposite direction that was happening in the mitochondria. So how do plants strip water of electrons and hydrogens? And let's just remind ourselves that just like the reactions within the mitochondria, the reactions here that are catalyzed by these various complexes within the thylakoid membrane are simply biochemical reactions. They're redox reactions in which we are catalyzing by photosystem 2 the transfer of electrons from water to PQ, plastoquinone. Here, cytochrome B6F is catalyzing the transfer of electrons from PQH to plastocyanin, PC. In photosystem 1, we are transferring those electrons from plastocyanin to ferredoxin. And in the ferredoxin NAD+ oxidoreductase, we're transferring electrons from ferredoxin to NAD plus to give us our NADPH. Two of these reactions release energy that is being used to pump protons into the thylakoid lumen. PS2 here, cytochrome B6F here. So the question becomes, we have two situations here where we are catalyzing a reaction that is going against reduction potentials. What do I mean by that? If you were to take and order the electron carriers of the electron transport chain in mitochondria, you would see that they are in the same order as in increasing reduction potential. Right? If we start NADH, that has a lower reduction potential than FADH2, which is lower than Q, which is lower than cytochrome C, which is lower than oxygen, and that's the reason the electrons move in that direction, constantly being sucked by a more electronegative molecule all the way until we get to the most electronegative molecule. Here's the order of electron carriers as they exist within chloroplasts, and what you will immediately see is that is not the same order as increasing reduction potential. In fact, there are two situations in which you're going from a higher reduction potential to a lower reduction potential. So here at PS2, if you compare the reduction potentials of water/oxygen and reduced/oxidized PQH, what you'll notice is water has a far greater reduction potential than PQ. So normally this reaction is endergonic. Same thing here in PS1. If you compare the reduction potentials of plastocyanin and ferredoxin, you'll see that plastocyanin has a far higher reduction potential than ferredoxin. So this reaction would normally be endergonic. What then are these reactions coupled to make them exergonic? And did you figure out sunlight? If you did, you'd be right. These two reactions, photosystem 1, photosystem 2, the ones catalyzed by these two enzymes, as the name suggests, photosystem, they rely on coupling the energy from sunlight to this normally endergonic reaction. And that's what's special about photosynthesis, about photophosphorylation. So let's take a look at what's happening here in photosystem 2. In photosystem 2, we're taking electrons from water, which holds onto them really well, and we're passing them on to PQ, which doesn't hold on to them very well. In fact, let's assign numbers to that. The redox potential of water is 0.816, and that of PQ is 0.14. Remember, the more positive the redox potential of a molecule is, the better it holds on to those electrons. And so you'll see that water is far better at holding on to those electrons than PQ. So what is it about photosystem 2 that allows this reaction to be exergonic? And the first answer is that photosystem 2, within it is embedded a special chlorophyll called chlorophyll P680. Okay, and P680 looks like this. It's got this big long side chain. That's what embeds itself into the membrane, and it's got this big part here that absorbs specific wavelengths of energy, of sunlight. And this, when it absorbs it, it has, or before it absorbs that energy, so naturally, it has a redox potential of positive 1.3. And remember, oxygen is 0.816. So this chlorophyll, which exists within photosystem 2, has the natural ability to steal electrons and hydrogens from water. It is naturally better at doing that than oxygen. It has a higher redox potential. So that's exactly what happens. Here, within photosystem 2, here's our P680. It can naturally, in an exergonic reaction, steal electrons from water, and that water then becomes, the oxygen part of it becomes O2. The hydrogen parts of it become protons that go into increasing the concentration of protons within the thylakoid lumen. Okay, So now we've got P680 that's got the electrons from water, and now that it has these electrons, it can't steal any more electrons from water until itself gets rid of those electrons. It has to get rid of those electrons, in fact, to the next carrier, which is PQ. The problem is, if P680 is so electronegative it's able to steal electrons from water, there's no way that PQ can steal electrons from P680. So when I ask you, what's the problem now? That's the problem. P680 is like the single highest redox potential molecule within plant cells. How is it going to then donate electrons to PQ, which doesn't hold on to electrons very well? Certainly not better than P680. The answer now is light. Finally, we get to the photo of photosynthesis, light. What happens here is photosystem 2 absorbs specific wavelengths of light, right? And specifically, it's P680 that absorbs these wavelengths of light. When P680 absorbs sunlight, it excites its electrons, and therefore its redox potential goes way, way down from somewhere around, 1.3 all the way to negative something, right? Negative, it looks like maybe 0.8 or so. So, when P680 absorbs light, it excites its electrons, it reduces its reduction potential very much, so much so that now PQ has a higher redox potential than this excited P680. Bang, now it can steal those electrons. That's why we need the light. The light has to be absorbed by P680, excites its electrons, reduces temporarily reduces its redox potential, and now it can pass electrons to the next carrier, which is PQ to PQH2. Now, if we take a closer look at photosystem 2, you're going to see all sorts of pigment molecules. These are called light harvesting complexes, and they're used kind of like a satellite dish, where P680 is the center of that satellite dish, and that dish itself are all these pigment molecules, or these light harvesting complexes that are helping to absorb the light. And then there is that transduction of energy to one light harvesting complex to another, and finally, boom, to the center, P680. So it makes the system more efficient. Okay, so now we have reduced QH2, and it's going to pass its electrons to plastocyanin, PC. For this reaction, you'll notice, look at this, if we call the red light as this squiggly blue line, and the light, just the light as this squiggly blue line, then notice only photosystem 1 and photosystem 2 are taking advantage of light, which means that this reaction, sending electrons from QH2 to PC, is an exergonic, naturally exergonic. And in fact, it's very similar to that of complex 3 within the mitochondria, passing electrons from Q to cytochrome C. And in fact, it's so similar in that it uses the Q cycle. PQ is equivalent to Q in the mitochondria. PC is equivalent to cytochrome C in the mitochondria. And so because you have that same problem of taking 2 electrons and trying to transfer them to something that can only carry 1 electron, you get that Q cycle happening. And just like what happens within the mitochondria, our cytochrome B6F complex also leads to the pumping of protons into, in this case, the thylakoid lumen. Excellent. So now we have reduced plastocyanin. And so now we go to photosystem 1. Aha, photosystem. We know that we're using photons of light, which means this reaction is normally endergonic, and we need to couple it with the energy of light, which means that our plastocyanin has a redox potential of 0.4, and our ferredoxin, FD here, has a redox potential of negative 0.42. So normally this reaction would never happen. Ferredoxin would never be able to steal electrons from plastocyanin. And that is why we use light again. And instead of having P680, what Photosystem 1 has is another special chlorophyll called P700. And like P680, P700 has a very high or relatively high reduction potential, about 0.4. And like P680, it absorbs a very specific wavelength of light, and when it does so, its electrons get excited, and its reduction potential goes way down, so here to negative 1.2, which means plastocyanin spontaneously, okay, and it's catalyzed by photosystem 1, strips electrons from PC, and then absorbs a wavelength of light, gets excited. When P700 is then in its excited state, it donates its electrons to ferredoxin, and that's catalyzed by photosystem 1 here. And then ferredoxin is naturally less electronegative than NAD+, and so our last enzyme of this complex, F-ferredoxin NAD+ oxidoreductase, catalyzes the exergonic reaction of transferring electrons from ferredoxin to NAD+ to give us NADPH. And this is the final step of the light reactions, as we see right here. Now this occurs within the stroma, and so that NADPH that we create here is happening, is accumulating within the stroma. So again, to reiterate, what's happening then at photosystem 1 is our PC is naturally stripped of its electrons from P700. P700 absorbs light so that now its reduction potential goes down. Then that allows photosystem 1 to transfer electrons from P700 to ferredoxin. Ferredoxin is naturally less electronegative than NAD+, and so NAD+ reductase is then able to catalyze this transfer of electrons from ferredoxin to NAD+, making NADPH. Here is a nice summary of what's happening. The light reactions need two bursts of light, one that activates or excites the electrons of P680, and one that excites the electrons of P700. So what we have now is two things. We have a proton gradient that has accumulated within the thylakoid lumen, and we have NADPH that is accumulating out here. So the next step is, well, let's use this proton gradient, this proton motive force within the thylakoid lumen. And the molecule that takes advantage of that is our good old friend ATP synthase. And so as those protons move down their concentration gradient from the thylakoid lumen into the stroma, ATP is created within the stroma. Be very careful, folks. Make sure you understand where that proton motive force is accumulating and where that ATP is being made. So what is the directionality of those protons when they move through ATP synthase? Okay, so now we have, after the light reactions, we have ATP that has accumulated within the stroma, and we have NADPH that has been made and is accumulating within the stroma. And now the next video will talk about the dark reactions, or the Kelvin cycle, or CO2 assimilation, and that is what uses the NADPH, that's what uses ATP.